Cloning describes a number of different processes that can be used to produce genetically identical copies of a biological entity. The copy material, which has the same genetic makeup as the original, is referred to as the clone. This is my victim, Eugene, and this is my lab assistant, Kyle.
Ethics of human cloning. Scientists believe cloning is biologically wrong, and it is morally repugnant. Most people are against cloning, and people are afraid scientists will control human life. There is thought of achieving mortality and high rates of birth defects. Religious factors, as Jewish people believe it is wrong and men were meant to reproduce sexually. Christians believe there's too much death and too much risk.
Process of cloning. This way of cloning requires two types of cells: somatic cells, which is what is being cloned, also known as the genetic donor. To get the somatic cells are usually obtained by a routine cell biopsy performed by a veterinarian. The other cell is the egg cell, which is collected by the female of the same species, also known as the egg donor. In the lab, a scientist extracts and discards the nucleus of the egg cell, which is the part of the cell that contains the egg donor's genes. The scientist then inserts the somatic cell from the genetic donor into the egg and fuses the two with electricity. The resulting fused egg contains the genetic donor's DNA. The scientist stimulates the fused egg cell, which activates the egg and causes it to divide just as an egg would if it had been fertilized by a sperm cell in a conventional reproduction. The activated egg is then placed in a culture medium. As the cellular division continues over the course of several days, a blastocyst, early stage embryo, forms. After about a week, an embryo transfer specialist transfers the blastocyst to a reciprocant female, sometimes referred to as a surrogate mother, where it continues to develop. After a full-term pregnancy, the recipient gives birth to an animal that is essentially the identical twin of the genetic donor.
Go.
Some pros of cloning are: it supplies organs for transplants, it allows infertile parents to have babies, it reverses aging, it can prevent heart attacks, and it can clean defective genes. Some cons are: it's dangerous, it leads to health problems, there can be mutations, it's unethical, risky, unhealthy, and not right. Let's try again.
Real uses of cloning in humans. Human cloning could provide genetically identical cells for regeneration medicine, and tissues and organs for transplantation. Such cells, tissues, and organs would neither trigger an immune response nor require the use of immunosuppressive drugs. Human reproduction would be benefited by cloning. There are ways to create a fertility treatment that allows parents that are infertile to give birth to a child with some of their DNA. Also, a plus in the cloning is that they can reverse the aging process. Also, cloning can prevent heart attacks by cloning healthy heart cells and injecting them into areas of the heart that have been damaged. Also, cloning can help keep the defective genes in someone. Everyone carries 8 defective genes which make people sick when they would be otherwise remain healthy. The woman with high risk of Down syndrome avoid that risk by cloning. Same with cystic disease, liver failure, kidney failure, leukemia, cancer, and can cure genetic diseases.
The purpose of this lab is to create a genetically identical clone of Mimi, a brown female mouse. The tools we will need will be a microscope, a petri dish, a sharp pipette, a blunt pipette, and a chemical to stimulate cell division. We will also need an egg cell donor and a surrogate mother to grow Mimi clone. The 6 steps are: isolate donor cells from Mimi and Megdo, remove and discard the nucleus from the egg cell, transfer the somatic cell nucleus into the enucleated cell egg cell, stimulate cell division, implant the embryo into Mimi, the surrogate mother, and deliver the baby mouse clone of Mimi.
Scientists that contributed to where cloning is today is Penentios B. Zaveros, Michael D. West, Joseph B. Sibili, Robert P. Lanza, and Bridget Goldsworth.